All right, welcome everybody to Module 10, where we are going to cover DNA and RNA. We're going to talk about the structure and function of DNA and RNA. We're going to talk about how DNA is replicated. Then we're going to go through the processes of transcription, so DNA being converted into RNA. Then we're going to look at translation, where RNA is translated into proteins and amino acids. And then we'll finish up with some other details about DNA and RNA itself. So in this first little video, we're going to really focus on DNA structure and replication. So for DNA, it was not known to be a component of cells um, until the late 1800s. But even before DNA was discovered and actually understood as being a part of the cell, other researchers and scientists kind of understood that there was this material that controlled the way organisms looked and behaved, uh, but they really didn't know what it was. And they really didn't understand kind of the role that DNA played in this process. And so, for example, we've already talked about Mendel a little bit, and the fact that he really understood, the processes of inheritance and laws of segregation and independent assortment. And remember, he called those factors um, heritable products, right? So really didn't know what DNA was. But by the early 1950s, there was a couple of discoveries that really led the scientific world, or brought the scientific world to the understanding that DNA is that hereditary material that is passing traits from parent to offspring. Um, at the time, though, what was not understood was the actual arrangement and form of the DNA structure. We really didn't have a good understanding of how the atoms were arranged and what exactly gave DNA its unique properties. So, why was DNA able to store information? How was it able to copy it? How was it being passed on from parent to offspring every generation? And so while we knew that DNA was this hereditary material, we really didn't have a good understanding of its structure, its function, and kind of how it worked in terms of information storage and replication of that information. Now, if we look at DNA in general, and we'll talk about RNA too, but the first part of this chapter is really kind of focused on DNA. But both of them, if you remember from our earlier module, DNA and RNA are nucleic acids. So they are organic molecules. And if you remember, the monomers of these nucleic acids are called nucleotides. And these nucleotides can make up the polymers, which are the long chains of nucleic acids. And so a nucleotide polymer is what we also refer to as a polynucleotide. Polynucleotides can be extremely long, and they can have any number of sequences of the four different types of nucleotides. So A, C, T, and G. So adenosine, cytosine, thymine, and guanine. And so you can have any order and any combination of those nucleotides in your polynucleotide. And an important aspect of these nucleotides is that they are actually joined together by covalent bonds, and that covalent bond joins one another, or it joins the individual nucleotides together at the phosphate and sugar groups of the adjacent nucleotides. And so you have a phosphate bound to a sugar, that's then bound to another phosphate, that's then bound to the sugar, so on and so forth. And so what you end up with is this sugar phosphate backbone. Okay, and so that's kind of what we refer to that structure in DNA. So this sugar phosphate backbone, and we'll look at an image of that here in the next couple slides. And what's also important about these nitrogenous bases, so they're not only connected to one another via these bonds, but they're also arranged like ribs that project outwards from that sugar phosphate backbone. And each nucleotide, like we again mentioned in an earlier module, consists of three basic components. So you have your nitrogenous base, and that base varies depending on the specific nucleotide it is. So for example, the nucleotide adenine will have that nitrogenous base associated with adenine, so on and so forth. And then the second component is that sugar group and then the phosphate group, okay? And where DNA really differs from RNA, so one aspect is that the sugar is different in terms of its chemical composition for DNA compared to RNA. So those sugars differ a little bit. And the sugar that is found in a DNA nucleotide is called deoxyribose. So that is why we refer to DNA as deoxyribonucleic acid. And if you compare that with the ribonucleic acid, or the nucleotide found in RNA, again, those sugars are slightly different in terms of their chemical composition. So here we have a figure showing the double helical structure of a DNA molecule. You have your sugar phosphate backbone. And so remember, one nucleotide is made up of that sugar, the phosphate group, and then the nitrogenous base. And remember that nitrogenous base can either be A, T, C, or G, so adenine, guanine, thymine, or cytosine. And that the adjacent nucleotides are bound to one another through those covalent bonds, where a phosphate group will bind to an adjacent sugar group. So for example, here you have a DNA nucleotide that contains thymine binding with an adjacent nucleotide that contains cytosine. And that bond is occurring at that sugar phosphate interface. And so you end up with this phosphate sugar alternating pattern that makes that sugar phosphate backbone. And then your nitrogenous bases kind of poke out like rungs on a ladder, or ribs that are kind of poking out from that sugar phosphate backbone. And this is going to lead to some interesting interactions, because it allows for specific interactions with other nucleotides that really end up forming that helical base structure that we see for the double helix. And this all comes down to the fact that the 4 DNA nucleotides, so your thymine, cytosine, adenine, and guanine, have these specific pairing rules. And so you will always have a single ring structure binding with a double ring structure. And so for example, A always binds with T, and G always binds with C in a DNA molecule. And so we can break these nitrogenous bases up into 2 different types. Again, your single ring structures and your double ring structures. So T and C, so thymine and cytosine, are single ring. Adenine and guanine are double ring. And you kind of see that in this image here. So adenine has 2 carbon rings, thymine has 1, guanine has 2 carbon rings, cytosine has 1. And that's also important because it keeps a general spacing between the rungs or the backbones of the double helix, right? So if you had like a 2 bases that paired up that were both double, then that area would be wider than, say, an adjacent area that had maybe two single ring structures. And so it kind of keeps that equidistance, but it also kind of sets some ground rules for what specific bases can pair together. And another place where DNA differs from RNA, so not only are the sugars different in terms of their chemical composition, but RNA also has one different base compared to DNA. So instead of thymine, RNA has uracil. So RNA is made up of uracil, guanine, cytosine, and adenine. Okay? Another important factor for RNA is that RNA is a single-stranded molecule, whereas DNA is a double-stranded molecule. Both of these molecules have that nitrogenous base structure. They have that sugar phosphate backbone. But those main differences, again, is the differences in the sugar molecules for DNA and RNA, so deoxyribose versus ribose. DNA having A's, G's, C's, and T's, and RNA having A, G, C, and U for uracil. And then the main difference too is single versus double-stranded. And so those are kind of the big differences between DNA and RNA molecules. Now, like I mentioned earlier at the beginning of this video, we really didn't understand the structure of the DNA molecule until about the mid-1900s, so around 1950, where work by James Watson and Francis Crick, along with data and previous information from Rosalind Franklin, led to Watson and Crick actually determining and figuring out the structural composition of DNA, and basically determining that DNA is a double helix or a double helical structure. So basically it's a helix with this uniform diameter, again, because of the always pairing of single and double ring structures that are made up of polynucleotide strands. And they came about this conclusion by visiting a laboratory of a researcher named Maurice Wilkins at King's College in London. And Watson basically saw an X-ray image produced by Wilkins' colleague, who was Rosalind Franklin. And so he saw an X-ray image of this DNA that was generated by Franklin, and then going on later and using that previous information based off of that X-ray image, him and Crick used wire models to look at constructing that double helix structure to basically go off of the data that Rosalind had, to basically confirm that data and what's known about the DNA. And so that led to the discovery of the double helical structure of DNA. So Watson and Crick were awarded the Nobel Prize. Rosalind Franklin had passed away, and so she did not get awarded the Nobel Prize because she had died before that award was given, which is a sad part of the history of the discovery of the structure of the DNA double helix. And so what we see when we look at the double helix of DNA is basically set up to where it looks like a ladder, a rope ladder, that is just twisted around its central axis. And so basically the sides of the ladder kind of represent the sugar phosphate backbones. So the actual, I guess you could say, vertical legs of the ladder, and then each rung that is attached to those vertical legs represent the individual base pairs that are connected to each other. And those individual base pairs are connected to one another through the formation of hydrogen bonds. And so these specific rules that I mentioned earlier about this base pairing really determines the side-by-side combinations that form the rungs on this double helix structure that is DNA. Here's a figure kind of showing that representation where we have a rope ladder. So this would be your sugar phosphate backbones. And then this would be your pairs of nitrogenous bases that are bound together by hydrogen bonds. And then if you kind of had an axis, right, if we just said, you know, you got an axis here, and then we just rotated, right, this ladder around that axis, which is what we're doing in the next two images, you get that double helix shape that we now know DNA exists in. And so if we look at a representation of DNA, we have three different ways to represent that. The one on the right-hand side is basically a computer model representation of the different atoms that make up those molecules. The blue is the sugar molecule, the yellow is that phosphate group, and then your different colors on the rungs are your different nitrogenous bases. Here you have what's known as the atomic model, where you're showing that phosphate group, the sugar group, the nitrogenous base attached to that, right? So that's a nucleotide. And then this nucleotide is bound to this nucleotide via a covalent bond. But then this base is paired with its appropriate base via hydrogen bonds. And those hydrogen bonds are these dotted lines here, okay? So you can see that G and C is connected by 3 hydrogen bonds, A and T by 2. And again, G is always going to pair with C, so guanine always pairs with cytosine, and adenine always pairs with thymine in DNA. And then this is just what's known as the ribbon model, where you basically turn the atomic model and computer model into a ribbon form, and you're showing those hydrogen bonds again with dotted or dashed lines. So again, in the 1950s is when Watson and Crick published their findings on the molecular structure of DNA. In '62, Watson and Crick received the Nobel Prize. And like I had mentioned, Rosalind Franklin had passed away before that. So in '58, she was only 38 years old. She had died to cancers. And so she was ineligible for the Nobel Prize because it's only given to living scientists, which is sad, because you know, if it wasn't for her X-ray, they wouldn't have probably made that discovery, at least whenever they did. So she was really instrumental for the finding of and determining the structure of DNA. And in their 1953 publication, the researchers wrote that the structure that they proposed, so this structure that's based off of these pairing mechanisms, suggests this copying mechanism for genetic material. So basically, DNA is this molecule that allows for an explanation. This is this molecular explanation for why we see unique properties of reproduction and inheritance that scientists have been observing, you know, for decades to a century before Watson and Crick actually discovered the structure of an arrangement of DNA. And what they also really looked at was that the arrangement of the DNA really affects its actions within the cell. And so we really see this relationship of structure to function. All right. And so that does it for our introduction to the structure of DNA and RNA. In our next video in the module, we are going to look at DNA replication and understand exactly how this copying mechanism in DNA works. So exactly how DNA is replicated. So see you in the next video.